Hello everybody and welcome to this little mini lecture on types of mutations. If you recall, there are two types of mutations, point mutations and frame shift mutations. In this mini lecture, we'll go through each type and see some examples of how mutations affect transcription and translation. Then next week, we'll talk about repair mechanisms that fix these mutations. But first, we'll start with the point mutations: insertions, deletions, silent, missense, and nonsense mutations. We'll start with insertion mutations. An insertion mutation is when one or more nucleotides is added to the sequence. Here we have our DNA strand, wild-type DNA strand, our transcribed mRNA strand, and our translated amino acid sequence. So in this example, we see that there has been a T inserted between the triplet of C's. In our wild type, we see this triplet was CCC, and now it's CCT, and the other C has been pushed down. This is an insertion deletion. A silent mutation is a point mutation that does not change the amino acid sequence. We see here that the last letter in the triplet that forms glycine is mutated from a C to a T during replication. This causes an A to be transcribed. You can see the A instead of a C. Instead of a G. But if we look at our amino acid chart, we can see that GGA still codes for glycine, just like GGG. So even though there's been a mutation and the base pair has changed, the amino acid that it codes for has not changed. That makes it a silent mutation. Now let's look at a missense mutation. A missense mutation is a point mutation that causes the newly transcribed mRNA to code for a different amino acid. In this example, we see the A towards the end of the sequence is mutated to a C during replication. This means that AGG is transcribed instead of AUG. So now where there should be a methionine, we have an arginine. A nonsense mutation is a point mutation that changes an amino acid to a stop codon. A nonsense mutation could also be created in that example we saw last week where the cytosine was deaminated and became a uracil. You remember that? Well, there's no way for RNA polymerase to transcribe a uracil, so that kind of change might interrupt transcription, even though it wasn't a stop codon. Here, in this nonsense mutation example, we see that the second nucleotide, G, is mutated to a T, changing this serine to a stop codon, blocking gene expression before it even starts. A deletion mutation occurs when one or more nucleotides is deleted or removed from the sequence. In this example, we see that the third nucleotide, the C here, is just removed. So the first three nucleotides are now AGG instead of AGC. Although this doesn't affect the amino acid, we can see that a serine is still coded. We can see that it does change the rest of the amino acids in the sequence. So instead of serine, histine, glycine, and methionine, we now have serine, methionine, glutamine, and methionine. That brings us to frame shift mutations. We've actually seen a couple of these in some of the previous examples. A frame shift mutation is when a mutation causes insertions or deletions that shift the reading frame. This reading frame shift to the left or to the right causes different amino acids to be translated, thus changing the entire amino acid sequence. In this example, we see that the sequence AGCGTA is mutated to be AGCTA, so it looks like a deletion mutation. And so the loss of that nucleotide changes the amino acid sequence. Instead of serine, histine, glycine, methionine, it's now serine, alanine, methionine, glycine. All right, so there's a little recap of what you've learned, read, and practiced on point mutations. So you can use this information now to go ahead and start your Pocket Mouse Lab Part 2. If you haven't already, it deals directly with these types of mutations. Otherwise, go ahead and go to your post-assessment and fill that out and continue on through the module.